All right, this is a walkthrough basically as an introduction and a general introduction to bacteria and how to take advantage of the information on microbiome prescription to better educate yourself on bacteria. And bacteria has a lot of information, and the site attempted to consolidate a lot of this information in a easier to access. You still may be facing a problem of having too much information, etc. One of the problems is that the typical bacteria test, we report anywhere from 500 to 3,000 different bacteria, which is probably a bit more data than most people are accustomed to handling easily. This basically allows you to dive into particular bacteria, which may be the ones of concern, the ones indicated as being too high or too low in the test results, and get some better information about it. Okay, so what I'm going to do is I'm going to cover the ability to search for a bacteria, and part of that is the issue of bacteria changing names, and we fortunately addressed that issue. So generally, even if the name has changed, we still can find by the old name. How to get general information about the bacteria, just general information. Then a chart showing what amounts are typically seen with the percentiles. The percentiles are much more meaningful than the amount. You may have 500 of a particular 500 out of a million of a particular bacteria. Now, is that too high or is that too low? It depends on which bacteria it is. When you go and convert it to percentile and you see that I'm in a 40th percentile, 40% have less and 60% have more than I, probably not concerned because that's sort of in the middle range. If you're sitting at the 99th percentile or the 0 percentile, then you need to take a look and see whether or not you want to change the amount of those particular bacteria you have. Then we go on listing medical conditions which have been the subject of studies which are associated with the particular bacteria. We will then move on to look at symptoms. And here I need to differentiate between conditions and symptoms. Conditions are generally defined medical conditions. Symptoms could be things like insomnia or having sensitivity to chemicals or brain fog. Often the conditions may incorporate those, but they don't focus on them in isolation. Our symptom analysis uses the approximately 5,000 samples which are being uploaded, many with symptoms annotated as in, okay, I have these symptoms out of a list of about 350 different symptoms, and people have annotated which ones they have, and because they have annotated it, we can do our own regression, identify which bacteria seems to be caused or seems to be associated with a particular symptom, so we know which bacteria are the ones you may want to focus on. Then we can look at suggestions that alter the bacteria, either increases or decreases it. If you have too much or too little of something, you want to know what can you do to help. Then we move on to bacteria that helps or hurts the bacteria you're looking at. In other words, a bacteria may produce natural antibiotics called bacterins. For example, Lactobacillus reuteri produces a one called ricin, which inhibits other bacteria. Other cases, bacteria produces a particular metabolite, like for example glucose, which other bacteria loves. So those bacteria that love it will increase more if this one particularly is present. Other cases, this one bacteria may decrease other bacteria. So we're going to look at what are associations there. And next we're going to look at the problem of filling the knowledge or study gaps by using inferences, using associations. And the problem is we don't have really that many studies. We have a very small number of studies, and most of the studies are with small sample sizes, and they don't really come up with a particularly high volume of data that is usable, which leaves us often facing with, okay, I have this bacteria which is too high, but I can't find anything in the literature on how to change it. That's our harsh reality. By using inference, we can get suggestions which are things that will likely help. Okay, let's go on and walk through these one by one. So ability to search is basically go to library lookup, and type in the name of whatever you're doing, and you go in, and what you will find is whatever name or part of a name you do, it will list things there. Notice the percent sign. The percent sign means a wildcard, anything like that before. So, for example, a whole bunch of different things shows up here. And two things are important to notice. One is the official name is the leftmost, older names are to the right, alternative names. And then over here we have the taxon number. The taxon number is a unique identifier which is consistent even when they rename bacteria, they keep the same number. And that number is the key to finding out if we are talking about the same. For example, are these all the same or are they not the same? Well, all these are the same. All of them have the same taxon number. So we can go in and look up, put in a name, hopefully check your spelling. It doesn't hurt to do a search on the bacteria you're looking for, because sometimes people have misspelled it, and sometimes lab reports have misspelled it. I've come across that more than once, which is real frustrating. So if you don't find something here, check the spelling by doing a general Google search and use the correct spelling. Okay, and what comes back is the official name, which is sitting there, and then the details with the NTBI taxon number and what type of bacteria it is. It will contain other information about, for example, if we know if it's a persistent colonizing species, and this link here to find literature. In this case, we also know it's found in the oral cavity in your mouth. So what is showing up in your gut comes from your mouth downward, which could be good or bad. And then if there are any studies of particular interest, we have them there. In this case, this is dealing with oral bacteria, and there's a link there. And then we have a whole bunch of different names, which this bacteria has been known. In some cases, they are interesting or comical, other cases not. And then we have general health information on the bottom about it, which gives general information, same type of information which you'll get if you ask about in ChatGPT, except you don't have to go all out and ask about ChatGPT. It's done for you already. The next thing is you see a nice chart, and the nice chart comes from a beautiful set of data which was shared with me, which has 1,000 samples of healthy people, and all of them done with shotgun, which is the most detailed way of getting a microbiome sample, and doing 10 million reads, which is the high end of the reads. So it's not the cheapest way, but it's the most accurate. And now you can go and see what the distribution is. So across up the side, you see what percentile. So the readings will go from 0 to 100 percentile for people who have this particular bacteria. And then you get the percentage of the sample. In this case, you see there's one person here which has 42% of their bacteria is this particular species, which probably means they have too much of that one. And if you go back here, you can go down and see all those too high. You really want to be probably down here at a 90th percentile or below, because once you get over 90 percentile, the numbers are looking very atypical. So this chart will often give you a visual indication. You can just go in and take a look at the percentage, go up and see where the curve line is. If you off the curve to the right, you are basically very high, and it always starts at 0, because generally a 0 report is seen just about everywhere, and that gives you an idea. So this chart is lovely. It is done with the best information which we have, and the most typical of what should be expected with a well done, detailed lab. Now, the next thing is listing conditioning association. This one is generally isn't shown unless you have logged in. So that's just simply to partially to prevent bots from ChatGPT harvesting data which is being assembled. So you can go here and see, okay, so we see for this particular one, bifidobifidum, we see that celiac disease, we have two studies found that it was high. COVID-19, we have 3 studies finding it low, etc. So you got high or low, and generally the studies only indicate if it's high or low, doesn't really give you any really good detailed information. So there's some challenges there, and elsewhere in the system when we're looking doing analysis of actual samples, we do a bit of creativity to convert these highs and lows into actual objective measurements. Next is symptoms. Symptoms are what people are self-diagnose, self-assert. And what we do is we go in and we look at the amount they have and see what it, and over here is chi-squared. The chi-squared is a statistical measure. The bigger than the chi-squared, the more significant it is. For example, low bifidobifidum is associated pretty strongly with general pain. It also associated with worsening symptoms with stress, etc. A high chi means that high levels are associated with it. So you can go in and look for it. There's a search box, and you can go and search for particular symptoms you want and see whether or not this is associated with it or not. So basically, this gives you a symptom association, like bloating, hair loss, etc. Some of these are studied, some of them are not, and it just gives you the numbers, and most of the numbers are based on biome site, which is a 16. It's not as accurate, but it is a, because we have a large number of samples with a large number of symptoms, it's a good match. The earlier chart was using a shotgun analysis only on healthy people, so we don't get associations coming from that. From here, we get associations. Next, we get to suggestions that alter, and we have it listed there. Generally, there are 2,000 items for a lot of bacteria, and you can go and you can censor it by particularly clicking in it to see what it studies. For example, we have Mediterranean diet, for which this one is probably bifidobacterium. 9 studies says that it increases it, and you can go through and you can say, okay, what food. You can go and do whatever. Synthetic impact isn't shown there. It should be showing up in a little while. I'll explain that later on in there. But this tells you how you can modify it. Now, if you have something high or too low, you want to do a holistic. You just don't want to focus on one bacteria. You want to say, okay, this will improve these bacteria which I am too low in, and then you want to check the ones you're high. You don't want to have the side effect that it also feeds the ones you are high in. So there's a balancing act. The algorithms being used on the microbiome prescription site does that all for you, but some people would like to do things themselves one by one, etc. And you can do it. That's the purpose of the site, is to make information available, where possible, guide you to what is probably, to the best of my knowledge and learning, the better or more probable suggestions, but you can go off and do your own thing. No problems with it. And the catch is you get a little icon showing a book, which means there are studies for it, and you will get the studies done. So you can go and read the study to see whether or not it does it or doesn't it. Okay, and we now go in, and below that, okay, when we go back to the chart showing the curve of percentage to percentile, at the bottom you will see something like visuals of protein interaction of bacteria of the same rank may appear on some pages it won't. And for example, this is a page it does, and we go and we see on the right, which will be on the top of the page, we see all the bacteria which are associated with it. And if you have high amount of this one, it will impact all those to be higher. If you have low amounts, it will impact all those to be lower. Okay, are you ready? You can see sort of the complexity starting to divide, because you may want to reduce one of these, in which case, how do you redo it? You reduce this one. Oh, okay. And over on the left, you have the number r squared is the amount of the variation explained, and we have something like 47% explaining for its impact on there. So in other words, it's a good there, and we see the degree of slope here. And if you're into statistics, you sort of can understand what it is, so it's there. Now, we also have the ability to click on the name, or over on the right, which is hidden, the parent, and get exploring, as you will see in the next slide. So we can click on the siblings and see what impacts the siblings, so you can start wandering across the whole microbiome, and you'll find that the items are shown in green or in red, as in the statistically you get a R squared of over 0.2, which is my threshold. You have to get at least 0.2, and the main reason is to restrict the volume of association to a manageable amount. And you can see what impacts it positively, and over here you can see what impacts it negatively. For example, bacteriolase has a negative impact on lactobacillus, which includes all your lactobacillus. So if you are low in this one, you probably want to reduce that one too if you can. So it gives you visuals of it, and remember, under all of them there is also a table. And by the way, as a further FYI, the thickness of the line is an indicator of how much the R square is. Bigger R squared, the bigger the line. So, and that's also reflected in the circles, as in bigger R square would be fatter, and the narrower circles will be lower. Okay, so let's go and go to the final tips and takeaway. One, the basic problem is for some bacteria, we do not find any studies showing what influences growth. Nothing, nada, absolutely nothing. But we may find that this particular bacteria is associated with a medical condition you have or a symptom you have, so you want to change it, but there's no study showing how to change it. So we try solving the problem by doing something called inference. And we have two ways of doing inferences. We can do a hierarchy inference, which is basically saying that take a particular bacteria, and whatever its parents is, whatever its children are, will probably, keyword, not guaranteed, probably have the same type of impact, but we probably want to diminish it. For example, if you're talking about a genus like Lactobacillus, its parent bacteria is probably going to be influenced in a similar way. Because why? Because it shares a whole bunch of the same DNA, so it's going to react similarly. The differentiation between them is caused by minor changes of DNA. So most of the DNA is going to be the same. So you're going to get them probably generally behaving the same. And the children bacteria will probably do the same thing. For example, Lactobacillus adelphi, Lactobacillus ariteri will all probably be influenced the same way. But we do a diminished one because we are not as sure or confident that that actually is going to happen. But it is a reasonable keyword inference. Now, the other one, which comes from what we just showed in terms of R squared and association, is association inferences. We infer for what changes its friends and enemies, again with reduced magnitude, what we should do, and I term that as a synthetic inferences or synthetic impact, because we don't have anything direct evidence of it, but we have the reasonableness of the suggestion because it diminishes its friends, and therefore if those friends are diminished, then chances are that bacteria will also be diminished. The nice joy about association inferences is that we can factor in the R squared factor and the slope in the inference. So the amount let's reduce the magnitude depends on R squared and the slope, so that we end up having a bit more accurate in our inference. Okay, so that's basically it. Lots of information, lots of diagrams to consider, and this is a quick high-level walkthrough. So that is who I am. I do have a Master's of Science. I can be reached at Research and Microbiome Prescription, and the site is microbiomeprescription.com. So I hope that this is a helpful, informative, quick introduction.